In 1928, a Scottish scientist named Alexander Fleming forgot to clean his Petri dishes before going on holiday. When he came back, one of them had grown mold, and the mold was killing the bacteria around it. That accident changed the course of medicine more than almost any planned discovery in history. Within 20 years, we had antibiotics. Within 30, we were using them to treat infections that had previously killed millions. The death rate from bacterial disease plummeted. We called it a miracle, and it was. But here is something Fleming understood that we seem to have forgotten. He warned about it in his Nobel Prize acceptance speech in 1945. Before the antibiotic era had even properly begun, he said that one day people would be able to buy penicillin anywhere, and if they took too little or used it carelessly, the bacteria would learn to survive it. That day arrived, and we were not ready. Think about the last time you or someone you know took antibiotics. A chest infection, a urinary tract infection, a course prescribed after surgery. You took the pills, finished the course, and recovered. That ordinary experience, unremarkable, forgettable, is one of the great privileges of living in the modern era. Before Fleming's discovery, a scratch could become a fatal blood infection. A ruptured appendix was almost always a death sentence. Childbirth killed mothers through bacterial infection at rates that are almost unimaginable to us now. In the decades that followed the discovery of penicillin, antibiotic after antibiotic was developed. Tuberculosis, scarlet fever, meningitis, syphilis, diseases that had defined human mortality for thousands of years, became treatable. It felt like a problem permanently solved, but in reality, it wasn't. In 2022, a major study published in The Lancet estimated that drug-resistant bacterial infections were directly responsible for 1.27 million deaths worldwide in 2019. That puts antibiotic resistance in the same mortality range as HIV and malaria for that year. The World Health Organization lists antimicrobial resistance as one of the top 10 global public health threats facing humanity. This is not a future problem. It is already here. Most of us carry an outdated image of bacteria. We picture them as simple, passive, primitive, small things that sit and wait. But bacteria are, in their own right, remarkable survivors. They have been on this planet for approximately 3.5 billion years. For context, multicellular life has only existed for around 600 million years. Bacteria predate plants. They predate animals. They predate everything we would recognize as complex life. Many fungi and soil bacteria have been producing natural antibiotic compounds for hundreds of millions of years as weapons against competing microbes. In turn, other bacteria evolved defenses against those compounds. This is the key insight that often gets missed in conversations about antibiotic resistance. Resistance is not a modern invention. It is ancient. Researchers have recovered resistance genes from Arctic permafrost samples dating back over 30,000 years. Long before a human being ever synthesized a drug, bacteria were already defending themselves against naturally occurring antibiotics in their environment. When Fleming discovered penicillin, he was not creating a new weapon from nothing. He was borrowing one from nature. And bacteria, as it turned out, already had partial answers to it. Let us follow a single bacterial cell to make this concrete. Imagine a single Staphylococcus aureus bacterium living on the surface of your skin. Under normal conditions, it causes no harm, but suppose it enters a small wound. It finds nutrients. It starts to multiply. Now, you take an antibiotic. The drug floods in. For the vast majority of bacteria in the wound, it is lethal. But in a colony of billions, some statistical variation is inevitable. One or two bacteria carry a random genetic mutation, a small copying error from an earlier round of division that gives them a slight advantage against the drug. Perhaps their cell wall is structured a little differently. Perhaps they produce an enzyme that can break the drug apart. Whatever the mechanism, they survive where the others do not. And then they do what bacteria have always done. They eventually divide. Under ideal conditions, bacteria like E. coli can double their population roughly every 20 minutes. Within hours, those resistant survivors have repopulated the colony, and the new population is, on average, more resistant than the original. This is natural selection operating in real time, not over millennia, just over hours. Researchers have identified at least 4 main strategies that bacteria use to resist antibiotics, and the most dangerous strains tend to use several of them at once. The first is enzymatic destruction. Some bacteria produce enzymes, specialized molecular machines that physically break apart the antibiotic before it can act. The best-known example is the beta-lactamase family of enzymes, which targets penicillin-based drugs. So many extended-spectrum variants can disable entire classes of antibiotics at once. The second strategy is target modification. Resistant bacteria can subtly alter the shape of that target so the drug no longer fits properly. Imagine changing the shape of a lock, so the original key no longer turns it. The third mechanism is called efflux pumping. Some bacteria have evolved pumps embedded in their cell membranes that actively push antibiotics back out before they can accumulate to a concentration that would cause harm. The fourth is reduced permeability. Certain bacteria simply close the doors. Their outer membranes contain protein channels called porins, through which antibiotics normally pass. By reducing the number of these channels, bacteria can prevent the drug from entering at all. Now, all 4 of those mechanisms are alarming enough on their own. But there is a fifth dimension to this story that makes the situation considerably more serious. Bacteria do not just pass resistance to their offspring. They share it sideways. Through a process called horizontal gene transfer, bacteria can pass genetic material directly to neighboring bacteria, including bacteria of entirely different species. The most common method is conjugation, where a bacterium forms a physical bridge to a neighbor and transfers a small, circular packet of DNA called a plasmid. These plasmids can carry multiple resistance genes. A single resistant bacterium can share that resistance not just with its descendants, but with unrelated bacteria in its immediate environment in the same generation. This is why a resistance gene that emerged in a hospital in one country can within months be detected in wastewater in another, in livestock in a third, and in gut bacteria of people who have never taken an antibiotic in their lives. Resistance does not travel only through biological lineages. It travels laterally, rapidly, and across borders that bacteria simply do not recognize. Nature gave bacteria the tools to resist antibiotics, but we gave them an environment in which to develop and spread that resistance at a speed and scale nature never would have produced on its own. The most significant factor is volume. We use enormous quantities of antibiotics globally. Research published in the Lancet Infectious Diseases suggests that between 2000 and 2010, global antibiotic consumption in humans increased by around 35%. Studies consistently find that a large proportion of antibiotic prescriptions in human medicine, estimates range from 30 to 50%, depending on the healthcare system studied, are either unnecessary or suboptimal. Antibiotics prescribed for viral infections, such as the common cold or influenza, have no therapeutic effect on the virus itself, but they do exert selective pressure on every bacterium in and on the patient's body. Every unnecessary course moves the needle incrementally toward resistance. But human medicine is only part of the picture. Agriculture uses antibiotics on a substantial scale. In some countries, historically, the majority of antibiotics by volume were administered to livestock, not only to treat sick animals, but also to promote growth at subtherapeutic doses. That low-level, sustained exposure is particularly effective at generating resistance, because it creates exactly the conditions under which bacteria can adapt gradually rather than being eliminated quickly. A 2014 study in the Lancet Infectious Diseases estimated that around 73% of all antimicrobials sold globally were used in animals rather than humans at that time. And then there is the environment. Antibiotic residues from both human excretion and agricultural runoff enter rivers, soils, and groundwater. Studies have found antibiotic compounds and resistance genes in river systems on every inhabited continent. Resistant bacteria thrive and exchange genetic material in these settings, and through wastewater, food supply chains, international travel, and direct animal contact, resistance spreads continuously across the globe. Now, here is something worth sitting with for a moment. Everything described so far, the bacterial evolution, the lateral gene transfer, the selective pressure of antibiotics, none of it was a surprise. It was the predicted mechanism. Scientists understood it from the start. Fleming spelled it out in his Nobel lecture in 1945. He described a scenario in which people could one day buy penicillin in shops. If they took too little, they would expose their microbes to the drug without eliminating them. Those microbes would survive, and they would emerge more resistant than before. He was describing antibiotic stewardship and the danger of misuse before most of the world had even heard the word penicillin. So the question is not why resistance happened. It was always going to happen. The more interesting question is what we did and did not do. The discovery of new antibiotics has essentially stalled. Between 1940 and 1960, a new class of antibiotic was reaching clinical use roughly every 2 years. Since the 1980s, only 2 genuinely new classes of antibiotics have made it to patients. The pipeline of candidate drugs in development has been described by researchers and health policy bodies as critically thin. The reasons are largely economic. Antibiotics are typically taken for short courses, 5 to 10 days. A successful antibiotic is one that clears the infection quickly and is then no longer needed. Compare that commercial profile to a drug for a chronic condition taken daily for decades. Health economists and pharmaceutical policy researchers, including those who contributed to the UK 2016 O'Neill Review on Antimicrobial Resistance, have argued that the current market structure does not provide adequate financial incentives for companies to invest in new antibiotic development. There is a further complication. Any new antibiotic that does reach clinical use is immediately treated as a last resort, held back, rationed, used as infrequently as possible to preserve its effectiveness. The result is a structural problem. The drugs most urgently needed from a public health perspective are among the least commercially attractive to develop. The consequences of this gap between need and development are already visible in clinical settings around the world. Carbapenem-resistant enterobacterals, commonly referred to as C, are bacteria that have developed resistance to carbapenems, which represent one of the most potent classes of antibiotics, available for severe gram-negative infections. When a patient develops a CRE infection, the treatment options can be extremely limited. Published studies report mortality rates associated with some CRE infections in the range of 40 to 50%. Methicillin-resistant Staphylococcus aureus, or MRSA, was for many years primarily a concern in hospital environments. Community-associated strains that can affect otherwise healthy people are now well documented. Drug-resistant tuberculosis represents a particularly serious challenge. Tuberculosis already causes around 1.5 million deaths globally each year. According to the World Health Organization estimates, of the new TB cases reported in recent years, approximately 450,000 annually involve strains resistant to at least the 2 most powerful first-line drugs. Treatment for multidrug-resistant TB can involve regimens lasting 18 months to 2 years, with significant side effects and with outcomes that remain uncertain even with optimal care. Many procedures in modern medicine depend on effective antibiotics to prevent and manage infection. Major surgery, cancer chemotherapy, organ transplantation, intensive care. As resistance erodes the reliability of that protection, the safety margins around these procedures narrow. This is not a theoretical concern. It is a documented trend that infectious disease clinicians and public health researchers have been monitoring and warning about for decades. Let us take a step back and look at what antibiotic resistance actually is at its most fundamental level. It is often framed as a failure of vigilance. Too many prescriptions, not enough investment, too little regulation of agricultural use. And there is genuine truth in all of those explanations. But there is something deeper running beneath all of them. Antibiotic resistance is evolution, operating in real time. It is natural selection, the same process that built the human immune system, that shaped every organism on this planet, working on organisms that reproduce in minutes, share genetic information across species boundaries, and have had 3.5 billion years of practice. We entered that arms race roughly 80 years ago. That is not a reason for despair. It is a reason for precision, because understanding the scale of what we are working against is the beginning of a more intelligent response. Some researchers are approaching the problem from a completely different angle. Rather than developing drugs that kill bacteria directly, they are working on compounds that disarm them, These molecules target the mechanisms bacteria use to cause harm without applying the lethal pressure that drives resistance. Early research in this area is genuinely promising, though it remains some years from clinical use. Bacteriophage therapy, the use of viruses that specifically target and infect bacteria, is receiving renewed attention. Phages were actually used medicinally before antibiotics were discovered and have been in continuous clinical use in parts of Eastern Europe throughout the antibiotic era. They are highly specific, targeting individual bacterial species without broadly disrupting the microbial community. They also evolve alongside their bacterial targets, which means they do not become obsolete in the same way a chemical drug does. In 2019, a patient in the United States received one of the first well-documented modern uses of phage therapy to treat a severe antibiotic-resistant bacterial infection. After all, available antibiotics had failed. Artificial intelligence is also beginning to contribute to the search for new antibiotics. In research published in 2023, a team used machine learning to screen large chemical libraries and identified a compound halicin with potent activity against several drug-resistant bacterial strains. These are early findings, not approved treatments, but they represent a genuinely new approach to a problem that has resisted traditional discovery methods. And the interventions with the most immediate impact do not require any of these breakthroughs, improving how existing antibiotics are prescribed only when genuinely needed at the right dose for the right duration. Strengthening infection prevention so that fewer infections occur in the first place. Continuing to reduce subtherapeutic antibiotic use in agriculture. These measures are available now, and the evidence base supporting them is strong. Now, let us return to where we began. That bacterium drifting in the opening of this video was not acting with intent. It was not targeting us. It was doing what life does, finding a way to persist. We are not, at a biological level, simply at war with bacteria. Our gut microbiome contains somewhere in the region of 38 trillion bacterial cells, roughly comparable to the number of human cells in the body. We depend on those bacteria for digestion, immune regulation, and processes we are still learning to understand. The relationship between humans and bacteria is ancient, intricate, and largely cooperative. Most bacteria are not our adversaries, but the subset of bacteria that causes serious infection, and that is evolving beyond the reliable reach of our current drugs, represents a challenge unlike most we have faced. Not because nature is working against us, but because we inadvertently gave an ancient, adaptive system exactly the conditions it needed to accelerate. That is a humbling thought. But humility here is not weakness. It is clarity. There is a quiet paradox at the center of this story. The more effectively we use antibiotics, the faster we erode their usefulness. Every infection successfully treated is, in a microscopic and statistical sense, a selective event. And yet we cannot choose not to treat a septic child, or a patient in surgical recovery, or someone with bacterial meningitis. The obligation to treat is too fundamental. So we find ourselves where most generations eventually find themselves, inheriting consequences that earlier decisions produced, and making decisions today whose full consequences we cannot yet see. The bacteria were here before us. In many forms, in many environments, they will almost certainly outlast us. The question is not whether we can defeat them. It never was. That depends entirely on what we do next.